Good afternoon all of you. I am Nana Gupta, DMLT student of DPMI. Today I am going to introduce a machine which is automatic tissue processor and it is used in histopathology lab. Basically, what is histopathology? Histopathology is a branch of pathology which deals with the diseased tissue. So, this automatic tissue processor is used for tissue processing. So, our first question in our mind is that why do we need to process a tissue? So, the answer is the tissue is smooth, soft and very fragile. So, if we cut thin sections for microscopy, we won't be able to do it. So, we have to provide a necessary hardness to the tissue to proper sections. So, in routine examination, we use paraffin wax to provide necessary hardness. So, if we are using paraffin wax, firstly we have to remove all of the water present in the tissue to make the tissue water free, as water is not miscible with wax. So, we treat the tissue in certain chemicals to remove the water completely. Basically, in automatic tissue processor, we perform 4 steps, that is fixation, dehydration, clearing, infiltration and impregnation. So, after grossing, we put the tissue piece in the cassette and a serial number so that we can identify the tissue. We can close this tissue cassette like this and put it in the basket. This is the basket and this is the top. The top helps the basket to attach to the basket rotors. We close this basket. Our basket is ready. Now we attach this basket to the basket rotors. So here is the lifting handle to open the machine. It opens in 2 ways. First position, then second. Now I am attaching this basket to the basket rotor. This basket rotor helps the basket to move one container to the other. Now I am going to close this machine similarly in 2 ways. This is our first step and this is our second step. Now I am going to tell you about the control panel of this system. First, it is the main switch, which it is the indicator that indicates that our machine has started on. This is the rotor switch and this is the indicator which indicates that rotor had started. The function of rotor is to rotate the basket in the container for proper mixing. Now our next control panel is program timer. Program timer helps to control the time of the program. Then there is the delay timer which helps to delay the tissue processing time. Here is the position display indicator which indicates in which our basket has been processing. After 1 hour each, the basket like this automatically ups and goes to the another beakers. Now in this machine there are 10 beakers and 2 wax bath containers. So here our first container is 10% formalin. Basically 10% formalin is a fixative and here in the container fixation process takes place. So basically what is fixation? Fixation is a process in which we preserve the tissue at its neutral state. And in both the containers, 1 hour each the process has been taken place. Next, after fixation, our process is dehydration. Dehydration is a process in which we remove water with the help of increasing concentration of alcohol, like 70% alcohol, 80% alcohol, 90% alcohol, 95% alcohol and 2 containers of absolute alcohol, that is 100% alcohol. Then after our dehydration process, our clearing process takes place. And clearing is the process in which alcohol is removed with the help of xylene and make room for paraffin wax during infiltration and impregnation. We commonly use xylene because it is miscible in both the solvents, alcohol as well as wax. After clearing, our next process is taken place in the wax bath. Wax bath has knobs which helps to maintain the temperature, and wax bath temperature must be between 50 to 60 degrees Celsius. As the temperature attains 50 to 60 degrees Celsius, the indicator indicates that temperature has been attained in the wax bath. In the wax bath, during infiltration, the xylene is eliminated into the surrounding tissue, into the surrounding melted wax, and afterwards the wax is diffused into the tissue, is known as impregnation. After overnight or 12 hours, the timer reaches its end point and indicate that our tissue has been processed. After the tissue has been processed, we have to push that red button. As I push the red button, the basket will come out of the wax bath and it is being processed. So we can take off the basket, and after tissue processing, we perform microtomy, embedding, microtomy, staining or mounting. And after mounting, our slide is ready for the microscope. So friends, if you have any question, you may ask.
May I?
Yes.
How do we know that dehydration is done or not?
For that purpose, we have to take 100% alcohol in a small test tube and add anhydrous copper sulfate. If the solution turns into the blue, that indicates our dehydration has done. Next.
How many times we have to change chemical?
You may change chemical twice a week. Next.
How many cassettes does the basket carry?
If the cassette is large, then it carry 20 in a basket, and if the cassette is small, it carry 35 in a basket. Next.
What is the main advantage of automatic tissue processing?
The main advantage of this tissue processor is it decreases the human error. Secondly, it saves the time and it gives accuracy in our results. Thank you and hope you have enjoyed this video.